smartlab lecture series. So today I will present a little the idea behind the smartlab network and the trends for collaboration. So the idea of this lecture series is to give a general overview of what each member or what the presenter is doing, followed by specific ideas of the or specific use cases from within his lab, which should trigger the possibility to find collaborative efforts. So to see to trigger collaborations within the network. So the history of the smartlab, the term smartlab is nothing we have invented here. So it's been used for a while, for at least a decade. One of the first prominent smartlab appearances in the field of lab automation was a smartlab in the originated from Hanover. Wait 1 second. Oh, what's it? Lab timer. From Dasha Beutel in Hanover, which has been presented in the Biotechnica in 2015 in the years afterwards. So the latest one this year has been organized from SmartLab Solutions in Dresden. Then there's also a SmartLab in different contexts. For example, the SmartLab company on e-mobility. And there also has been already a SmartLab Innovation Network, which was active from 2015 to 2019, which was trying to trigger organization collaboration within the community. So the idea was to create a community here. With our smartlab network, so we first want to define what smartlab means to us. So for us, smartlab, it means it's a combination of a smart digitalization, standardization, and automatization of lab works. And the aim of the smartlab, I can read this out, the aim of the smartlab network is to foster digitalization and automation in academic and industrial laboratories through the development of open and cost-efficient soft and hardware solutions. The network shall provide platform for collaborations, training and discussions between students, teachers, end users, soft and hardware developers. So, I mean, one of the main bottlenecks and one of the driving forces we see in especially in academic laboratories is problems with reproducibility and problems with interchangeability and documentation. And there are some societies which already are some efforts, some networks which already try to work on these shortcomings. One, for example, is the Society for Laboratory Automation and Screening, which are especially working on standardization. Then we've got two different societies which are also involved in the smartlab network, which is one, the Standardization for Laboratory Automation, SILA, which is focusing on standardized communication between laboratory devices. I deal with the same focus for the laboratory and analytical device standards, which has been sponsored by Spectaris. So why do we need another? Why do we need another conglomerate? And well, here we see one bottleneck where we come into play. That usually these societies, they focus on the existing automation strategies. So, for example, if one thinks about who's working or who's active in automation, usually we've got the trigger or the money comes from the user side, which is in this case as an example, big pharma companies. However, within the big pharma companies, they have specialized IT departments which run the automated systems and who are also in charge of setting up new systems. So for setting these up, the IT departments, usually they don't do it from scratch themselves, but they externalize this effort or outsource it to some integrators like external companies, which then develop the new software or hardware solutions together with the hardware providers, which then deliver the final automation system to the pharma companies to run it. So the standard or default players in lab automation, they are highly specialized and they are highly trained. So this is a well-established workflow. However, many of us work in an academic environment on a small and medium company environment. And if we look around in universities, for example, if we look for highly specialized and trained people, there's specialized personnel, but usually they are not specialized in automation. So for example, the group leader around me, they have a background in medical in biology and different science of pharmacology, but they are not specially automation or informatics people. Then what external companies do, they can externalize, they can just buy in the missing expertise. So this is, again, in academic environments, usually there's no funding to externalize these efforts. So it's very hard to get funding to set up an automation system. Same for the expensive hardware. Then another big caveat or problem in the academic environments is the high fluctuation of the people doing the research. So there are some permanent positions, but most of the people working in research labs, they are either students, master students, for example, who work doing maximally 6 months in the lab, or in the best case, PhD students working 3 years. But it's not that we have a very well-trained and staff working together for very long periods of time. The next point is that in basic research, the research requires a lot of flexibility. So not like in pharma companies where we have an established and certified production pipeline, which is then being run for another 10 years or so, and which is highly regulated. In academia, usually protocols vary quite a bit between one and another month or year or so. This leads to what is commonly known as the reproducibility crisis, that many experiments that have been done in one lab, they cannot easily be transferred to another lab, and that many labs have problems reproducing, for example, published results from other groups. Sometimes even results generated in the same lab by different people some years ago might create problems. So then for the academic environment, this idea of a lights out lab, where, for example, everything is fully automated, like total lab automation, and this is probably not what we are aiming for and what makes sense to aim for. And this is also because there are some examples, for example, when we start thinking about automation, there are examples of premature automation. So a lab, before it can start automation, it really has to be ready and set the framework for automation. And there's one recent example, which is not from the automation side, but which came, for example, from Tesla, when they, some 3 to 5 years ago, they initiated the production of the Model 3. They had huge problems in setting up the automation. Not because they're not capable of doing these things. I mean, they're capable to land a rocket on a small platform in the middle of the ocean. They should be able to do automation, but it shows how complex automation is and how many caveats one can find here. For example, as a general rule, flexibility is the inverse of automatability. So the more flexible the process has to be, the more harder it will be to automate it. And then second, which is a very good or important point, which I tried to stress in the last slide, if a process is not standardized, it is not suited for automation. So before we start thinking about automation and thinking about buying expensive equipment on robots, we should think about how to standardize the processes. And yeah, this is a bad. This graph, for example, shows the workflow in a more or less automated laboratory workflow. So on one hand, we can separate the flow of materials. So we've got the plates, we've got the samples, we've got all the labware, chemicals, what else? And the other flow is the data flow, which is here drawn in blue. And for a full automation, we have to automate all these 3 different flows. However, when we think about where to start, it makes a lot of sense to start to automate the data flow, because the scientists, they spend a lot of time gathering data, transferring data, and also the data gathering analysis. This is very prone of errors. So here, if it's done manually, there's always a risk of mixing samples. So this should be the entry point for laboratory automation. And when we think about standardization or standards for the digitalization, we can separate this into 4 different domains. So one domain for automation would be the physical dimensions of our samples. So I will show later. So one prime example is the introduction of the SBS plate format, that all plates being used have the same square format. Well, I've got some somewhere here. And this is, for example, what's been developed by SLAS, by the Society for Laboratory Automation. Then the second point of standardization would be standardizing the instrument control here on the top right. This is where CELA LATs are active. And here one example which works very nicely, which is usually used as a yeah, as a role model is the USB standard, which has been introduced in PCs and computers, and which really just enabled the plug and play capability, which we also try to get into the lab environments. And here to be more service oriented by having standardized protocols that we can mix and match machines from different vendors, and to have a vendor agnostic environment. Then on the bottom right, we see the standardization of data formats. This again, many machines, they produce even binary data, which cannot easily be read by generic tools. So here there are some groups, like for example, the Animal Society, which are working on standardized laboratory documentation protocols or formats, which can then facilitate and standardize the downstream processing, and also the logging. So this is one part of the documentation, is the logging of the sample, so that afterwards we can exactly follow what has been done to the sample at each point of time, which is, for example, in my case of live cell imaging, is or tissue imaging is quite important. The last part of this square would be the protocol definition. And here, this is, in my perception, also the, at the moment, least developed part of automation or standardization, that we should find a way to define protocols in a standardized fashion, so that protocols can be changed. And if a different lab is running a similar, not the same, but a similar hardware, they can just import the protocol and run it as similar as possible to the original publication. There are different approaches to this, like Python Lab or LabOP, but this is far from being a commonly accepted standard. And so this is again how the data collection could look like. So we've got a scientist having a manuscript, having a sample, running through the different machines, collecting data from all the machines, and at the end merging everything into a central storage where we can have some automated analysis system. In the labs we are looking at, which are mostly, well, very hand manual at the moment. This is, for example, here the lab in the beginning. This is a typical lab workflow we've got in the lab. This is, for example, media change in a sterile hood. So everybody can see this is quite manual written, no automated tracking of the labels and so on. So the question is, how can we get from this manual labor to these more or less fully automated systems? And buying such a system directly, obviously, would be too much of a jump. So our approach is to start with digitalization, and for automation, in any case, when we want to automate a process, we have to generate a digital twin of the whole process. And here we can, for example, start with a twin, starting with an electronic lab notebook, where we already start documenting all the results in an automated fashion, in a digital, documented, centralized fashion, where we can ensure that we've got backup of the data becomes searchable. So this is already a big advantage over paper lab notebooks. And then we have to see how we can get the hardware, like the real lab, the real equipment, how we can synchronize it with the automated or with the digital twin. And one thing is to do it, for example, using smartphone apps, where we can use the smartphone camera to have non-connected devices and to import data from non-connected devices. For example, in this case, by using the smartphone, taking a photo of the LCD display of the balance, taking a photo of the barcode of the sample, and then merging the data and importing it as a data set to the electronic lab notebook, which already in many cases would increase data safety quite a lot. So here you think, for example, here, these triangles, they should signify the required investment for the automation. So we see that these are legacy devices. They are probably already available in the lab. So this would require no additional investment. Cell phones, usually they're also available. And on the digital twin side, the same, like there are quite a number of free-to-use electronic lab notebooks. So here one could start doing digitalization in the first step in automation without any paywall, any barriers in terms of paywalls. So once this works, so this would be a single direction. So we would take the sample and the data would only flow to the ELN. So the next step of automation, one could think, for example, about Bluetooth-enabled handheld pipettes, which cost a couple of €100. But this would, for example, give the possibility to plan the experiment beforehand in a laboratory information system, a LIMS, and then the LIMS could, for example, transfer the required volumes to be pipetted to the pipette, pipette it, document the pipetting process, and write it to the electronic lab notebook. So this would be the second stage of data safety, again at a relatively moderate cost. And then the hope is that once laboratories start doing automation and they start to feel the benefits of automation, then it would be easier to invest in more sophisticated automation devices. And usually one of the first things that are automized is the liquid handling in the laboratories, because it's very repetitive, very tedious process. And there are quite a number of relatively low budget automation devices for this. For example, like here, this entry-level pipetting robot. So the second level, when we then keep going in the automation process, but sooner or later one needs to automate, especially if you, for example, if you have processes which run overnight or over the weekends, that is the plate handling. So basically in cell culture, what I do, it is the transfer of the plates from the incubator to a measurement or a pipetting device for media change or for analytics. There are different ways to do this, which I will go into further detail a little bit later. One here is a use of mobile robots, which can actually serve even devices which are further apart. And the other one is of just relatively simple SCARA robots, which can transfer plates from nearby positions. Okay. So just to recapitulate the different steps we've got here. So the first step, and this is the idea of the network here, to create awareness about automation, about the possibilities, about the caveats, what we have to take care of. What are the bottlenecks? So if I have a process which I would like to analyze in my lab, find the bottlenecks, identify the repetitive tasks, and then identify the low-hanging fruits. So we don't want to start automating the most complex parts of it first, but really start to, where it makes sense. So, and what, in my opinion, the first entry point one should think of is to introduce, if it hasn't happened yet, introduce documentation. So introduce electronic lab notebooks and electronic laboratory information systems, which is the basis of basically everything else. Then one can start to automate documentation. Can be done manual, can be done with the help of smartphone apps, for example, or by voice assistants. For example, here's a workflow which has been tested in our lab, where we can use smart assistants, smart devices to collaborate or to document our laboratory work hands-free with cloud service and withdraw data, log data, and help the researchers guide through the protocols without the need of paper notebooks. Then one would automate analysis by having automated analysis systems, automate liquid handling, which I've shown, and then the last step would be automated plate handling. This would be the last step towards full automation. So let's go to the laboratory notebooks. Yeah, this is empty. So when thinking about how to get to an automated lab, there are actually two scenarios which we have to distinguish. So one is the greenfield way, which is we plan a new laboratory completely from scratch. So we can completely redesign the infrastructure. We can make everything ready for automation. We can get the latest device generation, which are fully connected and everything. And we can already from the beginning take care of integration of proprietary systems. This is what happens if a big company decides to automate a specific workflow. However, in academia, usually what we have is what is termed here brownfield. So we already have an existing laboratory, and we have to see how we can transfer a manual laboratory into an automated laboratory. So we have to take care of use the existing infrastructure, which is usually built around humans, built for human use and not for machine use. We've got old lab devices which are often don't have integrated communication, and we've got very heterogeneous interfaces. So as I said, that when we order new devices. We can order devices which automatically have Zila or LATS protocols implemented. However, with existing devices, we just have to see how we make them connect, how to connect them. And here again, here are different ways, which I've been showing before. One is to have like legacy devices which just have visual interfaces, like LCD displays, which are not connected at all, which is, for example, here an old Casey counter. I mean, the good thing about these things is they are very robust, which has a downside that, for example, in our lab, we've got some equipment which is like 15, maybe even 20 years old, especially balances and in the Casey counters, for example. And here, instead of doing an electric automation or electric connectivity, one approach could be to use smartphones and to use smartphone cameras to identify the numbers of the displays and transfer them into a centralized repository, or the ELN. The second kind of devices is, for example, here these pipetting robots and Eppendorf pipetter. This again, they've been designed as standalone robots or standalone machines, which have been connected and started and stopped manually by a user. And if you want to connect them, usually they come with RS-232 communication or with some, like usually today, if somebody sees these kind of plugs or these kind of cables, many people don't know what to do with it because modern computers, they don't even connect to them. So here, another relatively cheap and easy way to make these devices communicative is using so-called Zila boxes or connector devices, driver boxes, where you've got a single board computer with all the serial connectivity required to talk to these machines. But on the other hand, they have Wi-Fi connectivity to then digitize the communication and send it again to the server in a standard fashion, making for a couple of €100, making these machines connective, which by other means would have resulted in buying a completely new device, which can easily add up to 40, 50, 60,000 euro for these pipetting robots. And the next thing in automation would be the plate handling. So there again, we've got two different ways to do the plate handling. One is to bring the device to the plates. For example, here, and this is one of the simplest setups I could imagine. So we've got an automated incubator where the cells or the plates spend most of the time. And then we've got a simple 4 degree of freedom transfer arm here, which takes the plates from the incubator, transfers it to an analysis station, here an automated microscope, and after measurement, brings them back. This is fair enough. This can still fit into a normally ordinary laboratory. However, when we come to more complex workflows where we've got different devices, different pipetting robots, and different incubators, for example, to get redundancy here, or to have cold storage also for chemicals, this gets quite complex. And what is often done is that we either have a robotic arm which is positioned in the center of the machine, being able to reach all different positions of the workflow, or the other possibility is to put a robotic arm onto a linear rail so it can serve different machines at the same time. However, what we see here, that when the robotic arm, for example, fails, some machine fails, or if you have a mixed process where some of the tasks are automated and other tasks are still done manually, it would be very complicated for a human to interact here with a machine. For example, just opening the windows would create a breach in sterility. And so another approach, which is not so common yet, but which I think has a lot of future, is to bring the plate to the devices as you would do it in a manual process. And here we've got automated laboratory self-driving robots, like for example this one here, which could co-work in laboratories which are designed for humans, and which, simply in the first step, could extend the working hours. So for example, the manual parts of the process could be done during the day by humans, and then the standardized parts could be done during the night or during the weekends independently by these machines. And now to be more specific, I would like to show you one use case which we are working on in the pharmacology in Göttingen, which is in this case a human-centric phenotypic drug screen, which this is our standard process, but we've got a specific one which we are going to execute next year, where we try to incorporate some of these automation approaches, which I've just shown you exemplarily. So the workflow or the drug screening, this is like a prototypical pipeline from the pharma industry, which most of you have probably seen many times. So in drug discovery, usually we start with a big pharma library with tens or even hundred thousands of compounds. And then we want to narrow down this big amount of these large libraries to the one compound, which finally become the commercialized drug. This is a very enduring, very long-term and very expensive exercise, costing over 1 or 2 billion euro and taking maybe 10 years in total. And the question is how we can make, first, where we can use automation and how we can make this process more efficient. And we see that in the classical drug screening and the preclinical screen. So these would be clinical trials where the medication is tested in healthy humans before it's being released to the market. And the preclinical ones, many of the experiments, especially the safety experiments, they had still done in rodents or in animals. And here our approach in the pharmacology department is to go away from animal models and to do a completely human-centric drug screening. So this can be done either with human cells, like human two-dimensional cells, like as here, or single cells, or with human organoids. For example, this is our main workhorse. It's an engineered human myocardium. It's a human model for heart tissue, but we also have human, we call it BINO. It's like brain organoids. Here is a schematic view. Here we've got the imaging view, and we also have fused models like this one, where we actually fuse the neuronal tissue with the muscle tissue. So the good thing is that we work completely with human cells from A to Z. So, and we can make tissues which are cohort specific. So, for example, if you have a sick or a specific genetic disease, we can create all this tissue and all these organoids from this one patient and from this one cohort, and have more realistic outcomes. For example, there are many diseases where there's no animal models available simply. So in this case, we have a two-step screening approach. So on one end, we've got the human. This case, for example, a sick, like a diseased human, somebody with a genetic defect, that we can take from the patient or from a representative patient. We can take a sample. We can dedifferentiate the cells, have induced pluripotent stem cells. Then we can amplify the cells to get large amount, in this case, for example, of cardiomyocytes. We can differentiate them to cardiomyocytes. Then we would have a large fast screen on a 384 or 1500 well format to do a quick and rough screen. And then from the best candidates from this pre-screen, we would then rescreen them on cardiac tissue organoids to finally get this one important drug. And this is the way how we plan on doing this in the pharmacology department. So 2 years ago, we acquired this high throughput confocal microscope from Yokogawa, which is completely automation ready. So we've got a pipetting robot in here, which can be seen in the microscope. This we are using on 384 well plates, where we can seed roughly 10,000 cells per well, and then do relatively fast, relatively high throughput screens. You see here next to the microscope, we've got an automated incubator. So when we want to do the measurements, we have to shuttle the plates from the incubator to the microscope. And then here you see a 3D view of the room. So opposite of the microscope, not like here in the photo. We have a sterile workbench with a pipetting robot that would then do the media changes. So at the moment, this process has been by hand. So we've got a scientist who takes the plates, analyzes it, does a media change, and brings it back. So for a full automation, we could do 2 or 3 different things. So one thing would just to bring all the machines closer together, so we can have one central robotic arm in the middle, which can then shuttle the plates, which is not what we want, because then we reduce the flexibility to do the process by hand. The second one is to get a big robot, which has the same reach to go across the different machines, which has been scheduled here, but which is again not practical and also not safe if we then work in parallel with people. So, and what we are going to start next year, or beginning of next year, is we're trying to do what is being done manually, one-to-one by one of the automated robots. So getting one of these Kevin robots, and then just all day driving in the triangle between the incubator, microscope and the pipetting robot. Another thing which I will not go into details here is, of course, the digitalization of the image analysis. So this microscope produces terabytes of data, and we already have more data than we can analyze. And one key factor here is really that the analysis pipeline has to be fully automated. So this is how our cells look like. We've got carymyocytes, and they are pre-labeled. So this is we've got a knock-in carymyocyte where the sarcomeres are GFP labeled, so we can see the sarcomeres which are actually generating the force in the carymyocytes. And we've got some automated analysis protocols, pre-trained neural networks, which can identify the sarcomeres, identify the sarcomere lengths and different features of the sarcomeres, the orientations, the quality of the orientation, and have automated analysis. So without this automated data analysis pipeline, the whole hardware analysis would not make sense at all, because we would just create much more data than one could possibly analyze. So automation should always start with the data automation and with the data transfer automation. So the second part of the process is, which is what we've most experience in, is the work with three-dimensional engineered human myocardium, or EHM, which is small three-dimensional tissue rings. So these rings, they are grown on a 48 well plate, which we also developed here. Here we already see that also from the plate format and also from the number of cells we need per plate, that the throughput is much lower. It's about 1/10 of the, or more than less than 1/10 of the automated 384 well format. And it's also, being more cells and more incubation times, it's also much more expensive. So we really want to have a two-step process where we do a pre-screen to filter from thousands of compounds, filter a couple of dozen, which then go into the more detailed high-quality screen here. So the tissues, they take a 4 week of maturation, which is a standardized process where we basically have daily media changes and quality measurements. And then we've got 1 week exposure to the drugs we want to test. So here's the example, for example, how the casting process, how the generation of the tissues looked before we started the automation. So this is a classical protocol which has been developed almost 20 years ago, where a researcher does by hand pipetting the cells into circular molds, and then by hand transferring them to these maturation devices to mature them for a couple of weeks, and then again by hand transfer them to measurement devices. So it's quite obvious that this gives nice tissues, as you see here, but it's by no means a basis for automation, because these steps would simply not be able to, or would be very hard to automate. So one of the first things we did is we generated a plate protocol, these 48 well plates, where we can cast and grow these tissues in a standardized format, which is being seen here. And by having a standardized format, we can just use ordinary laboratory robots or pipetting robots, for example, in this case, to cast the tissues and also to do the media changes, which is, as you see here, it's a very repetitive step. So we've got 48 wells being media changed every day. And yeah, so this would be a standardization of the physical dimensions of the plate. And then in the next step, we come to the automated handling of the tissues of the plates, which is being seen here. So in the front end, you've got the incubator, which can't be seen, and then you've got a robotic arm which grabs the plates, in this case taking it from the pipetting robot and unlidding it. And here, for example, this would be one example how this robot could and generate the tissues. So instead of having a pipetting robot, we can also use this 3D robot to cast the tissues. And then afterwards, you put the lid back on and bring it back to the incubator. And okay. Yeah, this is a video. This is a video on the analysis process, and this is what the setup looks like. So this is for example, this is a custom-made sterile hood we've built around this pipetting or this industrial robot in the middle. And here we've got the cell hotel, the cytomat, the incubator. One thing we've added here is when you look into the CAD, that we've installed the analysis, the measurement device. We've actually installed it inside the incubator, so that for the measurement under cell culture biologic conditions, we don't have to remove the plate from the incubator direction. So the analysis are being done here, and then the plate is being taken out only for the media changes. So yeah, as I said, this robotic arm, for example, is an industrial robot. It's quite heavy, and we didn't have a choice but to build a custom hood around it. However, in terms of standardization, in terms of spreading automation within our network, we've decided to redesign the whole equipment and to make it more lightweight and more standardized, more compact. And here's, for example, one approach we've started where we're using a standardized laboratory hood, which is in this case a 1.20 meter hood. And we would install, instead of having one industrial robotic arm, install 2 collaborative robotic arms. So the nice thing about collaborative arms is that they are very lightweight. So they are not dangerous for people interacting with them. And secondly, which is nice here, they don't generate so much vibration. So you've got more flexibility in mounting them, and then can be mounted in a standard sterile hood without having to reinforce the whole frame of it. And this is one of the approaches one of the ideas of the smartlab network, to completely construct and develop this automating work cell and then offering it as an open source hardware component to the community. So people trying to go into sterile automation, they can duplicate or build this machine if they have a good workshop, or order it from an external company for a relatively low price, because these companies, they don't have to invest in the development of these things. And by being open source, it's also very flexible. So the main ideas that we have here for different tasks, for example, this one would be the incubator again for our very specific engineered tumor myocardium. But then we have got flexible choices with the end effectors, that you can have end effectors for plate handling, but you could also have end effectors for 3D bioprinting or for organoid transfer, so that depending on the specific focus of the laboratory, what could easily adapt this machine or this generic machine to the needs for the specific experiment. And so this was quite a rush through the program. But of course, this has been, especially the second part of it, has been on the development in the Department of Pharmacology and Toxicology in Göttingen, which is from Professor Zimmermann here in the back of the photo, and which is a relatively large department. So we've got people working on cell reprogramming, cell amplification, tissue engineering. We also work on engineered hard patches for implantation for GMP processes. So it's quite an extent. So the lab automation is basically only a minor part of the whole department. And much of this work has been done in a long-standing collaboration with the Department of the Institute of Mechatronic Systems in Hanover, the EMS, especially with Leon Budde. And this is a photo of the department in Hanover. And yeah, with this, I would like to thank you, and I'm looking forward to answer your questions.